Hi, my name is Clara, and today I'll be talking about the growth dynamics of epizoic Achnanthes elongata and non-epizoic Psammodictyon panduriforme in co-cultures, as this is basically a poster presentation for the Molecular Life of Diatoms. It's going to be brief, and let's go. We know that diatoms are omnipresent in nature, especially if there's water, we can find them there, and that includes marine animals, big vertebrates, especially sea turtles, in our case loggerhead sea turtles that we study. And what we observed is that loggerhead sea turtles carry a lot of specific diatom taxa that isn't found anywhere else yet, at least, and diatom opportunists, the taxa that you can find everywhere else, but they're also on the turtle. And what we were wondering about is what makes the epizoic taxa specific. Why are we finding them only on the turtle in numbers high enough to be detected almost every time, consistently, even though they are competing all the time with all the opportunist species that grow much, much faster? We are able to isolate these diatoms in culture, and we notice that they tend to grow usually slower than the feistier taxa that are not epizoic. So the questions that we wanted to test them out by growing an epizoic diatom and a non-epizoic diatom together to see how they interact with each other on a very basic way. So what I did is I grew our diatoms of interest in monocultures. This one is Achnanthes elongata, a diatom that we consider to be epizoic for now. In recent research, it's been found on many different turtles all over the world, and it's very, very consistent. And let's say these diatoms here, Psammodictyon, the first strain that we used, and the second strain, we find them even in the surrounding water, maybe the water tanks where the turtles are being held, so we don't consider them epizoic at all. So what I did was I grew these in as single diatom culture, and I mixed Achnanthes with the first Psammodictyon strain and Achnanthes with the second Psammodictyon strain in two different conditions, grew them in 24 well plates over 19 days, imaged them every second day, and that's how I got a lot of images that needed to be analyzed, artifacts removed, different glitches deleted, and after that I could work with the numbers. This is how it looked like. So the Achnanthes is usually quite long. Those cells can be huge, but the issue with it was that it could also grow vertically since it attaches to one of the sides of the cells to the bottom and then just swims vertically in the media. That made it harder to even detect it automatically, so I couldn't automatically count it. I had to manually check the images for these kind of cells to differentiate them from the competitors. Well, after I did that, I cleaned the data, and the first thing that I got, this is still being analyzed, but these are kind of first insights into those numbers, the growth curves. So the dark kind of brownish color is the monoculture of Achnanthes, and these lighter are the Achnanthes in co-cultures. The orange one is with the first Psammodictyon, the small strain, and the second one is with the second Psammodictyon strain, the bigger cell that we saw on the previous slide. What we could notice was that even in the 19 days, the monoculture and the first co-culture, Achnanthes, did not reach stationary phase, but the Achnanthes in the second co-culture with the bigger Psammodictyon, it started reaching stationary phase around day 13, as well as its competitor. So these blue guys are Psammodictyon, the first strain, monoculture, co-culture, and here it's the same based on the legend that you can see on the side here. When I actually showed the growth, the maximum growth division per day like this, the only significant difference that I could find was between the Achnanthes monoculture and the Achnanthes in the second co-culture. So we realized that there is something going on there that maybe makes the Achnanthes cells grow faster, at least initially in the beginning, maybe a bit later. Should grow them longer to see the effects, maybe in bigger wells so they don't overcrowd each other. Maybe these effects are because of nutrient limitations, we're not sure yet. The important points to consider in this case is that we use non-model diatoms. The Achnanthes diatoms are very sticky. in comparison to the non-epizoic strains that we use here. This kind of approach system, the co-culturing just two diatom strains, they were xenic, so they carry bacteria with their culture, so the bacteria of the two mixed. But still, from this we can get very limited information. We know that multi-species biofilms are very complex in their interactions, so this is very, very reduced, but still it provides the first insights about how epizoic diatoms could be different, and if other diatoms are kind of helping them stay on the turtle and grow more efficiently. What we also noticed is that these two Psammodictyon strains, even though based on morphology they could be the same species, when we looked at the marker genes, this is just some initial data, it could seem that they're two different species, which could explain the different effects that they have on Achnanthes. Well, thank you for watching. For more details, just go to my poster. If you have any questions, just let me know. I'll try to answer them. Thank you.